Okay, if you'll recall, we're reviewing the three phyla of algae that have multicellular members. And we've already looked at Phaeophyta, the brown algae, and Rhodophyta, the red algae. So now we're going to take a look at Chlorophyta, the green algae. And chlorophyte algae are an important group for two reasons. They really show the evolution of multicellularity, and they also have very close common ancestry with plants, as you might imagine. And one of the things that is evidence of that are the photosynthetic pigments that they have. So they have chlorophyll A and B, and then accessory pigments that plants have too, like carotene, which is orange, and xanthophyll, which is yellow. And I know that's kind of hard to see. Sorry about that. It's trying to be fancy, making it yellow. And together, these pigments absorb the blue and red ends of the visible spectrum and reflect green, which is in the middle. And that's why they're green algae, and that's why plants are green. Chlorophyte algae live in marine, freshwater, and wet terrestrial habitats. So they live all over the place. Saltwater, freshwater, and even in puddles. If the puddle's there long enough, you might find green algae growing there. They're most closely related to plants, as I pointed out, and the evidence for that is their pigments. They have the same pigments: chlorophyll A, chlorophyll B, carotene, xanthophyll, cellulose cell wall, multicellularity, some of them, alternation of generations, which is something that we're going to be looking at 2 slides from now when we look at the life cycle of ulva, and DNA sequence, and especially DNA sequence. So the most modern and impressive evidence that green algae are most closely related to plants is by looking at the nucleotide sequence of their DNA and comparing the nucleotide sequences. And that's true for a lot of species. When we want to know how closely related species are, these days we go to DNA sequencing, because DNA sequencing has become relatively easy to do, and it doesn't take as long as it used to. And it's been found that the group within the chlorophyte algae, known as Charophyceans, so this is class Charophycia, are considered the most closely related to plants. And that's this photo represents a Charophycean algae. This is underwater, and you can kind of tell by the bubbles in the photo. But they're named after the stonewort Chara, and that's what we're looking at here. And if you were to see this in the water, you might guess it's a aquatic plant, because some plants do live in water. But it's actually a multicellular chlorophyte algae. So how did the evolution of multicellularity happen? Well, it's colonial organisms were like the middleman, like a stepping stone to multicellularity, and I hope that makes sense to you, or at least it will by the time I'm done explaining. So here we have Chlamydomonas, a unicellular algae, right? So unicellular organisms are composed of only one cell, unicellular, right? So Chlamydomonas is a good example of that, a unicellular algae. Colonial organisms have relatively few unspecialized cells that live together. So if you'll recall from our lab, gonium is a colonial algae, and each one of these cells that are living in a colony, a gonium colony, are very Chlamydomonas-like. So it's pretty clear and easy to see the evolutionary relationship here between this unicellular chlorophyte algae and this colonial chlorophyte algae. So it's a small colony of cells. Volvox, if you'll recall Volvox from our lab also, it's a large colony of cells. So one thing that makes that makes something multicellular is the number of cells. So you see here an increase in the number of cells making up individual organisms. Chlamydomonas here is an individual organism, has only one cell. Gonium is an individual organism made up of a few cells. Volvox is an individual organism made up of many more cells, still considered colonial, however. And Spirogyra here is a filamentous colonial organism, so the cells are in chains. Multicellular organisms are composed of large numbers of specialized cells, so many more cells, many more than Volvox, for example, organized into tissues and organs. The key word here is specialized. So a large number of specialized cells that are organized into specialized tissues and specialized organs. Specialized. So colonial organisms are colonial because they have relatively few cells and there aren't very many cells that are specialized, and those cells that are specialized really aren't together into specialized tissues and specialized organs. Whereas in multicellular organisms, specialized cells are together in specialized tissues and specialized organs. Ulva is considered a multicellular algae, but it's a relatively simple multicellular algae. And then Chara here, Charaphecian algae, is a very complex multicellular algae. And again, it's considered to be most closely related to plants. Of all the chlorophyte algae, the Charaphecians are considered most closely related to plants. So the idea is that multicellularity arose as colonial species evolved specialized cells, tissues, and organs. So we went from unicellular to colonial, and then colonial organisms evolved specialized cells, tissues, and organs to produce multicellular organisms. So this arrow represents the direction of evolution, from unicellular through colonial to multicellular. That's the idea. And I hope that makes sense to you. Okay, we're going to compare the life cycles of a unicellular chlorophyte algae and a multicellular chlorophyte algae. And here we're looking at the unicellular, and hopefully you'll recognize this little guy as Chlamydomonas. Chlamydomonas reproduces sexually when conditions are unfavorable. So notice that the life cycle diagram here is divided up into two halves, one being sexual and the other being asexual. And so Chlamydomonas is going to reproduce sexually when conditions are unfavorable. And that makes sense because it's going to produce a zygote that has this thick protective wall around it so it can survive unfavorable conditions. So in the sexual life cycle, individuals that are already haploid will produce gametes. And notice that the gametes are labeled plus and minus. They're opposite mating strains, as we refer to. And because that parent cell is haploid, it can produce gametes. It can produce a gamete by mitosis. So we don't need meiosis to reduce the number of chromosomes because the number of chromosomes is already in half, right? So they can just produce gametes by mitosis. And then this is a gamete from another individual. So this individual produced the plus gametes, and another individual, like this is representing from another life cycle of another individual, produced this minus or opposite mating strain gamete. And they come together at fertilization, fuse together their nuclei, and that produces a diploid zygote. So fusion of plus and minus gametes, fusion of the opposite mating strains produces a diploid zygote. The zygote then grows a thick protective wall that can survive conditions that would otherwise kill it, and it will not germinate. It will stay in that condition, dormant, for a long period of time until conditions become favorable. And so we would call that a zygospore, because it's a spore that contains the zygote. So this makes sense, again, because it's dormant, resistant. It can survive unfavorable conditions for a long period of time. And the other reason it makes sense for Chlamydomonas to reproduce sexually when conditions become unfavorable is the general result of sexual reproduction. Remember, sexual reproduction shuffles genes between two individuals. Again, these two gametes came from two different individuals, and they're going to fuse together and shuffle their genes together to produce new individuals that have variation. So this produces variation and would allow Chlamydomonas to evolutionarily adapt to changing conditions, because variation is the raw material of natural selection and evolution. So since the zygospore and the zygote is diploid, these spores the release of these zoospores, or these zoospores are produced by meiosis, because notice we're going from diploid to haploid. So the life cycle of Chlamydomonas is mostly haploid. You know, the whole thing is this tannish color in the diagram, which is haploid, so mostly haploid. The only diploid is the zygote and the zygospore. It immediately, or when it's time to germinate anyway, it will undergo meiosis and return to the haploid condition. So we go from the full number of chromosomes back to the half number of chromosomes, and that's how Chlamydomonas, the adult Chlamydomonas, is haploid. Chlamydomonas will then reproduce asexually when conditions are good, and that's to increase their populations to take advantage of good conditions, of plenty of light availability and nutrients and so forth. And they do so by multiple fissions. So notice that we're going from one individual and then through mitosis, asexually producing 4 individuals. So there has to be more than one binary fission to go from 1 to 4. So that's referred to as multiple fission. But it's still by mitosis and cytokinesis, just like binary fission. So Chlamydomonas gametes, and notice here that the Chlamydomonas gametes and zoospores look like miniature adults. So that's just an interesting observation. It would be as if, you know, like if this was the same in humans, it would be as if our gametes look like little miniature people. Be kind of funny. So that's the life cycle of Chlamydomonas, representing a unicellular green algae. Now we'll take a look at multicellular. So the life cycle of a multicellular algae, and in this case it's Alba, is very different from what we just saw in Chlamydomonas. And it's because it shows this life cycle known as alternation of generations. And it occurs in certain algae and plants, and that's a significant thing. These are chlorophyte algae, and they're thought to be most closely related to plants. So it makes sense that the life cycles would be similar between these multicellular algae and plants. They both show alternation of generations. And we're going to summarize what alternation of generations is doing here in the life cycle, and then we'll come back and take a look at some of the details. So there's an asexual diploid sporophyte plant. That's what we're seeing here. And notice it's diploid. It's in the blue of the diagram. So it's a diploid sporophyte. And it's best for you to always refer to it as that. Don't just call it a sporophyte, call it a diploid sporophyte. And that'll help you remember what's going on here in alternation of generations. So this diploid sporophyte generation, and notice I'm trying to emphasize that, that's a generation, produces haploid spores. So here we're producing haploid spores by meiosis, right? Because this is a diploid sporophyte. The only way it can produce haploid spores is through the process of meiosis. The only way you go from diploid to haploid is through the process of meiosis. So here we've got spores, and then the spores are going to develop and germinate and develop into haploid gametophytes. And that's another generation. That's the next generation. And those haploid gametophytes then go on, and again, we're just kind of summarizing now, those haploid gametophytes go on to produce diploid sporophyte, a diploid sporophyte. So they're going to mate, sexually mate, and produce gametes to sexually fuse together at fertilization and produce the diploid sporophyte. Notice that that's happening by mitosis, because these gametophytes are already haploid. So they can produce gametes by mitosis. There doesn't need to be, and really can't be, any reduction in chromosome number. So to produce haploid gametes, these haploid gametophytes can do that through mitosis. We then get a zygote that's going to become or develop into the diploid sporophyte. So the sporophyte is diploid and produces haploid spores by meiosis. right? So that's what you want to remember. It's a diploid sporophyte, and that's why it'll help you remember that it's a diploid sporophyte. And it's producing haploid spores. The only way you can go from diploid to haploid is by meiosis. So that's what you want to remember. And then the gametophyte is haploid. The gametophytes, whether they be male or female, are haploid, and they produce haploid gametes by mitosis. So we're going from haploid to haploid, so there's no change in chromosome number. So that's carried out by mitosis, always, if there's no change in chromosome number. And then those haploid gametes fuse together to go back to diploid. And remember, the only way you go from haploid to diploid is by fusing two haploid cells together, and that's what's happening at fertilization. So there's no, and the weird thing here is that there's no visual difference between the diploid sporophyte and the haploid gametophytes. You can't tell by looking at these, at least without a microscope, that this is a diploid sporophyte and these are haploid gametophytes. So it would be as if your parents are haploid, and remember that humans, the body cells, all your body cells are diploid, but this would be as if your parents were haploid and they produce you, and you are diploid, and then you're going to produce, your offspring are going to be haploid, and then your grandchildren are going to be diploid. You know, it doesn't work that way in us, but that's what's going on here. The generations are alternating between diploid sporophyte and haploid gametophyte. Alternation of generations. And it would be good for you to kind of recite this for a while. Diploid sporophyte, haploid gametophyte. Diploid sporophyte, haploid gametophyte. Diploid sporophyte, haploid gametophyte. Diploid sporophyte, haploid gametophyte, just to kind of drill it into your brain of what's going on here in alternation of generations. And then just an interesting side note here that kind of suggests something about evolution. Notice that all the spores and gametes, the spores here and the gametes, look very similar to each other and to Chlamydomonas on the previous slide. They're flagellated little unicellular organisms look like, but they're not. They're gametes and spores, but they look like Chlamydomonas. And this suggests common ancestry between the chlorophyte algae. In other words, ulva here, which is a chlorophyte algae, is, you know, this suggests that it's pretty closely related to Chlamydomonas, which is a chlorophyte algae. And that multicellularity has unicellular evolutionary roots. And that's what we were looking at a couple slides ago, how we went from unicellular through colonial to multicellular. So here we're looking at the ecology and human use of algae, and the ecology of algae is that they're very important producers. And when I say producers, I mean I'm referring to primary productivity, which is the same thing as photosynthesis, the rate of photosynthesis. So that's what this diagram is about, or this graph is about, this bar graph. Primary productivity, the photosynthetic rate. Photosynthesis releases oxygen, right? So the open ocean, the pelagic zone, and that would be the photic zone of the pelagic zone. In other words, only the part of the open ocean where light penetrates contributes mostly to global primary productivity. So the open ocean, right? Notice the bar for the open ocean is much bigger, or not much bigger than the tropical rainforest, but it's big. So a huge contribution to global photosynthesis and oxygen production is carried out by algae. And more specifically, phytoplankton, those unicellular and colonial algae that are floating around in the open ocean. Over 50% of the Earth's oxygen. And I think this is like the third time this has been pointed out in our notes. So that's an important contribution of algal species. Another ecological importance of algae is that whole concept of eutrophication and cultural eutrophication, which is a negative thing that's going on in freshwater habitats. But that's not on this slide. We'll be looking at that some more coming up. But how do humans use algae? Well, you can see here, you know, we eat it. You directly eat some algae. You know, if you eat sushi, a lot of sushi is wrapped in algae. But we also get derivatives from algae. So like if you look on a label of ice cream, you might see carrageenan listed. It's and alginate. These are algal derivatives, molecules that are taken from algae and used to make foods thicker and creamier, like ice cream. Also found in cheese and chocolate milk, and peanut butter and pudding and frozen desserts, fruit drinks, a lot of other foods. So even if we don't directly eat the algae, there are a lot of algal derivatives that end up in our food. And then other algal derivatives are used in products like paint and the agar that we used back when we cultured bacteria in petri dishes. So it's used, algal derivatives are used in industry a lot. So that's it for our review of the different phyla of algae, and or the plant-like protists, also known as the plant-like protists. We're going to continue on, because algae live in aquatic habitats. We're going to continue on looking at aquatic habitat ecology, and that will wrap up this unit.